I think pretty soon it will become malpractice to not be using an AI sort of co-pilot for a diagnosis, because the odds are you'll miss something. And we shouldn't let anecdote and sort of one individual guided medicine trump the power and accuracy of a large aggregate sum of data. Imaging-based methods, there's an old joke that you can give 10 slides to 10 pathologists and get 11 different diagnoses. This is the old issue with when you look at how do you take a slide with H&E staining or other stains and say, okay, well, what is this? Is it infiltrating? Is it not? Is this a tumor? Is it a pre-tumorous? Is it, that itself has gotten better over the years, but it's still problematic. But what's really a panacea that's coming in is just machine learning and the artificial intelligence tools that can mine all the image data. And so by definition, they can be a better co-pilot than grabbing 3 of your favorite pathologists down the hall to help you interpret a complex image. You grab the aggregate wisdom of millions of pathologists and tens of billions of data sets, and then have that be your guide. So again, you're not going to replace the doctor, but you'll just augment essentially what the knowledge base and the experience of the doctor. So it's not anecdote-based medicine, it's precision medicine because it's precise, it has enough data to be precise.
Welcome, Dr. Mason. I'm so excited that you can join me on the Augmented Life.
It was a pleasure to be here. Thanks for having me.
All right. Well, one of the reasons why I've been looking forward to talking with you was that when we had Scott Kelly on the show, he talked about the effect on telomere length while he was in space. This just kind of blew my mind. Now, I had known just from the previous work that we had done together before he came on the show about that telomere effect, but something new that came out when we were doing our interview was that it returned to normal later once he became earthbound. This kind of blew my mind. Like, I was not expecting, it's always interesting when physical forces show reversion to the mean, which I think of as a statistical phenomenon. Now, it's been several years since you were the principal investigator on that study. What have you learned since? Just let's talk telomere length.
Yes, yeah. So telomeres, for if anyone in the audience doesn't know, is they are kind of the caps at the ends of your chromosomes that contain on the DNA for your molecular code for genesis of your body in response to life and disease. And so it's kind of like shoelaces that protect the end. The little caps of your shoelaces that protect your shoelaces and make it so they don't fray away. The same thing is true of our DNA and on our chromosomes, is that it's protected by and really packaged nicely and maintained by these telomeres. And they shrink as we normally get older. So shorter telomeres are on average in people that are older. So if you see longer telomeres, you should be younger. And so that was what was so surprising, is we thought the stress of spaceflight, the radiation, and this was work with Susan Bailey's lab as well. She got the first results in the study, and then we validated them in our lab, and we did additional sequencing methods in my lab to try and really tease this out because it seems so surprising. But it's been validated now multiple times for the twin study, working with the Bailey lab and our lab, and also in Commander Kelly. And then also we've now seen it in 11 other astronauts. And then we just published recently the Inspiration 4 crew. They were in space for only 3 days in orbit, and that was enough to begin the lengthening process as well. So it is a very rapid phenotype of spaceflight where the telomeres seem to get longer and they seem to be very adaptive within days, right? So this is what was surprising. It wasn't a matter of months or even he was in space for a year, Scott Kelly, but really quickly. So we've now seen in almost every astronaut we've looked at, this seems to be a consistent feature, which is really interesting.
Right. Any speculation on the mechanism of action? Like what is triggering this?
Yes. So there's been a lot of work in the past 5 years to tease that out, as well as, but basically, it's happening, but why? So interestingly, the two biggest causes are likely, is it the radiation or is it the microgravity? It could be other things like, stress, it could be immune perturbation. But the two biggest changes to the body, of course, are the radiation and microgravity. And we've now done tests in cells on Earth. If you just irradiate them, you can also get longer telomeres. And we also see mechanistically a non-coding RNA called TERA, T-E-R-R-A, that's activated, which goes right to the sites where the telomere, where there's double strand breaks, and we see that's also lengthening telomeres. So we know that the radiation is likely the biggest cause, and we know that the molecule that seems to mediate it is called TERA is involved. And we can also recapitulate this on Earth. We can do it in Plasmodium falciparum. We published that. Radiation seems to be the biggest driver, where it is a response to the damage, and it also kills off the cells that were the most likely to die. So it's both killing the weaker cells that were probably about to die anyway, plus the radiation response is what we think now.
Oh, how interesting. Ooh. Oh, so this is a different mechanism of autophagy than like other kinds of stress like fasting.
Right, right. Yeah, kind of a way to cleanse. Now, they also the term for is like hormesis. If you stress the body, you can get some aspects of longevity primed effectively. But it is, a little bit of stress on the body can be a good thing. One of my favorite examples of this is actually at our cancer center at our hospital. Some of the radiation therapies actually prime the immune system by doing a lower dose of radiation before doing immunotherapy, with the same idea is that you don't want to completely destroy the immune system, although you can do that for, say, transplants, but you want to prime the immune system and kind of activate it, which we've seen with radiation, which is interesting.
Wow. One of my own sort of personal obsessions is trying to collect the most longitudinal data about my life that I can, with the idea of having baselines to compare things to. So if something goes wrong, it'd be really nice to know what it looked like 10 years ago. And so I have this, I think of it as like a folk practice because it's hard to be rigorous about this, of conscious hormesis. I think of it as my hormetics practices. So I will do multi-day fasting, cold exposure. I'm not really sure. So there are two challenges about this. One is, I guess I just have an open-ended exploration that, I guess my fundamental theorem is that exposing yourself to some acute stressors might have long-term health effects distinct from like chronic stressors, which seem to do, the opposite. So, let's just periodically just see what this body is capable of. It's just kind of the principle. But how do you how do we decide on periodicity? What's the minimum effective dose? I have no idea.
Well, and so you're going in the right direction because there's a lot of evidence that some level of this hormesis and stress on the body has a cleansing effect, has a priming effect. A great example is just even people when you lift weights. If you just do the stasis, you won't build muscle. But if you push it a little bit farther than you normally have pushed it, then you'll build muscle. If you go too far, you'll break a bone, right? So there's always this space. You have to push it just a little bit, just enough to get a phenotype change. And that is something we've seen with marathon runners, weight builders, athletes. And but to your point, there haven't been large scale systematic studies of if I do a cold plunge, is it 30-second cold plunge, 1 minute, 2 minute, 5 minute, 10 minute? What's the temperature? How much is it a cofactor of my age or body fat or how much you just ate? There's a lot of cofactors that go into this. But I think, and I think we all long for something that's better advice than when you get to the doctor and they say, well, okay, just go work out more and eat more vegetables. It's generally like the most common grounded suggestion because it's generally true. But I think we all want a little bit more granularity, something you could do that's not even a one pill that makes it easy, but give me more specificity about exactly the sort of what we do with medicine. We do precision medicine. We guide the right treatment to the right cancer patient, to the right dose, to the right drug based on the genetic profile. Like that. So we're just beginning to do it now. What I'd say wellness and precision wellness is a term that one of my colleagues has popularized. We've talked about recently in my recent book.
Right. So this is something that I'd love to get into with this, let's say, theory of the case. I'm really enthusiastic about precision medicine. It seems to me like we are on track for some extraordinary new norms in healthcare. Let's say 20 years from now. If you'll grant me that, if 20 years from now there's scaled precision healthcare, it's mass personalization, let's say,
and not just sick care, not like when you get
sick,
but
right?
Like where actual healthcare, where it's when you keep you're healthy and keeping you healthy, not just waiting until you get sick and addressing it then. Yeah.
Yes, yeah, I'm looking forward to exactly that transition. But in advance, what is it that we can do as individuals to add instrumentation to our bodies or start doing some data collection that our future selves, let's say 20 years from now, will be like so happy that we collected those baselines from our past selves?
Good question. So I think the ways we can begin to use molecular tools today and genetic tools to monitor are really just emerging. So to take a step back even for a second, this was really impossible until we had a reference human genome, which wasn't until 2001. And it wasn't cheap enough to do whole genome sequencing or really at scale profiling until the last 6 or 7 years. So if anyone is listening and they feel like, oh no, did I miss the boat? Like, no, the boat just got to the dock. Like, we haven't been able to do any of this kind of work at scale or at a cost that's reasonable until the past few years. And so some of the things you can do, though, is one of them is a multi-cancer early detection test, or MCED, you'll often see it's called, is a way, and Grail is probably one of the most well-known examples of a company that's looking for early detection of cancer in your blood. And Freenome is another one. Tempest does this. There's a wide range of companies that do this kind of work, but you want to look for fragments of DNA in the blood that just indicate a mutation that's different from the rest of your DNA, that indicates it's either from a tumor or a risk factor for getting cancer. But ideally, if you catch it early enough, it actually is from the tumor, but when it's just stage 1 or stage 2, and you catch it early instead of stage 3 or stage 4 where it's metastasized all over your body, which by definition it's easier to detect because you have more cells that are basically exuding these fragments of DNA that carry the mutation. But if you catch it early enough, you could then detect it and say, okay, let's confirm it and then treat it right away. And then you're treating a tumor the size, of a dime instead of something the size of a softball. So this is much easier surgically, therapeutically, recovery-wise, all those factors. So that's one thing that I'd recommend. There is still a lot of tools for epigenetic aging or biological aging, and Nathan Price I was referring to earlier, and Joel Dudley, Paul Jacobson. We've created companies and sold companies that do some of this, and so there's a lot of activity in this space, some of which I've directly been involved in. But it's very early days. So if your epigenetic age, or when you look at DNA methylation, or how your sort of chromatin and your regulatory state changes, ideally having a younger epigenetic age is better, but we don't know how much that matters yet, or how well that it translates into a long-term view of your mortality risk. These are some big unknowns, but those are things that are worth looking at, I think, today. There's also microbiome profiling. You want to see, do you have a good, healthy gut microbiome? The microbes in your skin and your gut, they also can, they're not just there, and you want them to be diverse and robust and have good keystone species, but certain species like Akkermansia, for example, mucinophilia, they regulate blood glucose. And so if you have a good sort of gut ecosystem, it makes it so you don't have these big spikes in your glucose, and that'll decrease your chance of getting diabetes. So some of these things are not just human cells, but the microbial cells, and really thinking long-term how the body regulates blood glucose, how it regulates sort of its structure of its gut and its metabolism. These are all factors that really can help your longevity and your overall health stay well.
Well, it strikes me that one pattern in the recommendations that you just gave is how, let's say, quantitative they are. And I'm going to set up this contrast. I mean, you might just reject this premise, but indulge me for a moment. I was very enthusiastic about new startups offering things like full body MRIs for no particular reason. Let's just scan the whole thing and see what we find. Then my enthusiasm waned after actually doing some experimentation here, because I guess I had a mental model of MRIs as being very, I'll just say, science-y. It seems like, wow, this is extremely advanced technology. And it was only after I pursued this that I learned, wow, there's a lot of interpretation going into this. Is almost like a sense impression more than like we are extracting genetic material and we'll tell you if there are tumors in your bloodstream, right?
Right, and tell you the exact mutations and their exact location at the frequency of that mutation with really unparalleled accuracy. So it's a great point, actually, because imaging-based methods are, there's an old joke that you can give 10 slides to 10 pathologists and get 11 different diagnoses. It's an old issue with when you look at how do you take a slide with H&E staining or other stains and say, okay, well, what is this? Is it infiltrating? Is it not? Is this a tumor? Is it a pre-tumorous? Is it, that itself has gotten better over the years, but it's still problematic. But what's really a panacea that's coming in is just machine learning and the artificial intelligence tools that can mine all the image data. And so by definition, they can be a better co-pilot than grabbing 3 of your favorite pathologists down the hall to help you interpret a complex image. You grab the aggregate wisdom of millions of pathologists and tens of billions of data sets, and then have that be your guide. So again, you're not going to replace the doctor, but you'll just augment essentially what the knowledge base is of and the experience of the doctor. So it's not anecdote-based medicine, it's precision medicine because it's precise. It has enough data to be precise and accurate. So I think It will get better. It is early days on the imaging, and you can actually, there's some places and I'm in Brooklyn right now, and there's places down the street where you can walk in and get a full MRI and pay out of pocket, and they'll look for things. So I think that's still a little bit on the early stage, but that concept, I think, probably within a year or two, it's not that far away. Well, they'll have generated enough data, and the machine learning tools are that good that they're already as good or better than most pathologists. So again, I'm not saying we should replace most of the radiologists or pathologists, but I think it'll be pretty soon what would become malpractice to not be using an AI sort of co-pilot for a diagnosis, because the odds are you'll miss something, and we shouldn't let anecdote and sort of one individual guided medicine trump the power and accuracy of all, the large aggregate sum of data. So
Yeah, yeah. Implicit in that interpretation, there's a certain sense in which we can build a model that is doing judgment well, right? This interpretability. There's another layer in which I'm seeing AI make improvements in real time, which is simply image segmentation. Like the one of the things that made the connectomics research on a relatively small amount of neural tissue so powerfully good is that there was a machine learning segmentation model that actually like picked apart individual elements of glial cells versus this sort of axon versus this. And so at the level of, that's almost like a perceptual advancement. At the level of distinguishing microstructures, you have the computer do it. So then the humans can do the much more powerful like inference on top.
yeah, they're real. They're almost like additional researchers in the lab. They help find things and guide you. And I think and people think of generative AI taking over humanity and being our overlords. And maybe someday that'll happen, but maybe they'll be helpful overlords. Maybe they'll take care of and organize resources and be beneficial, or it could be, the Terminator movies and they destroy us all. But I think if we program them well, they'll be helpful, and they already have been. So I think there's a great, there's many positive examples where the AI tools have been really instrumental or essential to a new discovery. And so I think it's the tip. It's just getting started, and I think I'm very optimistic about where it'll go.
I'm optimistic too. But part of the reason why we named this content series The Augmented Life is that I'm looking forward to like machine plus human augmentation, which I think leads to better outcomes than either achieve on their own. Literally, the best chess games in the world right now are sometimes what's called centaur chess, which is you get grandmasters, so you get truly amazing chess players, but they also get to use a computer, research their next move before they make it, and computer plus grandmaster outperforms even the best chess computers, as well as, of course, the best grandmasters. So it's some of the most advanced games in the world.
Well, it's interesting for imaging-based diagnosis for cancer. There was a paper just last week that showed that's usually what I think our intuition would be, is that grandmaster plus AI would be better. You get the best of both worlds. But there are cases where if you have that sometimes the human's intuition will have this memory of something that was impactful to that person, but is not statistically more likely. And in some cases, that the machine, it's better without the human, as these papers have started to come out now.
Oh, I know, I saw that. Like it got major publication. I think that might have been in Nature, right? Like, yeah. So the the unassisted AI did better.
Because in principle, no, it has to be trained correctly, and the training data, the machine's only as good as the training data. And people say, our AI algorithms, are they racist? It's like, well, no, they're just a machine, but the data set was racist or it was biased or both, in terms of what was fed to it. So if you feed, racially biased data into a data set, the algorithm will just mirror that. So and so that means society has structural issues that what it's collecting or what it's feeding versus the machine. But the machine will do what it's taught, and even build upon that. So it is, and it can be more powerful for imaging diagnostics than a physician who, again, by definitions, maybe seen thousands or tens of thousands of images, but not billions. And so it's just, but you always want someone to double check it and sign off is what you want, and say, well, just to sign off. But at this point, it's only like the machine that is there only to check to make sure there's nothing that would be of harm to the patient. Because at some point, you would probably no longer be able to question the diagnosis of the machine, because it's probably statistically, if we show it more accurate than the human, you wouldn't defer to the human. You just use the human to just double check it, basically, which is what we do now.
Yeah. I'm walking around with a deep neural net up here, and I'm like on guard for overfitting. Like I'm pretty sure I have over extrapolated from what is essentially just anecdotes all the time.
But we're getting better, and it's hard. We're humans. I mean, I view this as a chance to build a human brain type structure that is on guard for and maybe overcome some of those limitations of our current brain structure. So, um, but people get afraid. They feel like, does that mean? I actually have met multiple people in the past year who are just despondent, and they don't know if they should. They don't know what to tell their kids because they feel like anything you can do in AI can do better. Like if it's everything from image analysis to writing poetry to writing a novel, like it can do whatever you want to do. It can do it better. And so it's like I don't know what to tell my kids because they've got no future. And I say, oh, they just might have an easier future. But he was really terrified.
Wow. Wow. Well, I happen to think that it's like emergent phenomena all the way down. Or maybe the better way to put this is it's emergent phenomena all the way up. I think consciousness, my own experience of consciousness is probably some emergent phenomena of some evolved number of processes happening in my brain. But that's the same for a functioning society. And so depending on the level of abstraction that you care about, we already live in a world in which, for instance, society can do everything that you can do except better.
Right, right. And machines can do anything you can do physically, but better. Like I'm not going to be able to lift a giant boulder, and that's okay. That there's a machine that can lift a big boulder and build giant buildings and go to space, right? So that's okay. That it is a collaborative effort with them. And I think people's fear is just that the intelligence will then view us as a threat at some point and take over. And Elon Musk and many others. But I think anyone who is afraid of that has not programmed enough because that means if that occurs, we did a really bad job of programming. We did an awful job of programming. So.
Well said. All right, so let's jump back on the optimism track. I the I You gave me a lot to follow up on in terms of new diagnostics or data gathering that I can do myself and set up a practice of. I love the idea of early detection of cancer. Any idea, speaking of, actually, there may be some rigor that we can apply here. So is there some understanding of, for instance, growth rates that imply an ideal period at which you should do your early cancer detection, right? Like obviously once every month is ridiculous, right? Because the rate of expansion doesn't, there's no benefit to that, right? And so I don't know, maybe once every 20 years is too long. Can we try to bookend this?
I think it's a good question. I think, yeah, probably will end up being annual for a lot of them, but and of course the short answer is still we don't know exactly if you're. It's interesting because for a lot of diagnoses, for example, there's a burst of risk for a lot of diseases that is in basically your 40s, 50s, and 60s, and then if you can kind of make it to 80, 85, a lot of them start to go away. Like there's this really critical period of cancer and other diseases, cardiac disease. And if you can kind of live through that, your risk of a lot of diseases go down. But you just have to get through that critical period. So I'd say in that frame of, let's say 35 till 70, probably every year, and maybe even between 45 and 55, I'd say you could even do twice a year. If you could, assuming it could be, it assumes like money's no object, which of course doesn't happen. But if we could get it, so the Grail test, for example, they funded a large study with the National Health System in the UK to follow hundreds of thousands of people to see if they could track early cancer. And they're paying for the study with the goal that if it looks really good, then everyone would get this done once a year as soon as they hit age 35 or 40 and keep tracking. So it's in their interest, of course, to prove out the value of their asset, the Leary assay. But I think we're all hopeful that it will be, because then it would mean you have a surveillance system that's tracking early cancer. So I think probably annual would be my gut reaction now, but it depends a little bit on how old you are.
Yeah, got it.
And also some things, it's also without indication. So if you do suddenly have a symptom or a phenotype, like, I feel like something has gone wrong, or I'm suddenly bleeding out of my ears, you might want to get checked.
Right.
Something has changed. So you should go into the doctor.
I've also been I've been collecting microbiome data at the level where things are getting sequenced. So I'm getting a pretty good idea of very, very specific types, at least of bacteria. and population sizes can be, I think, reasonably well extrapolated from my gut microbiota. Um, Going back to practices of hormetics, I was thinking, like I'm going to add deep, Korean style skin scrubs to my regimen, maybe like twice a year. And I've been wondering, I wonder how this affects my skin microbiome, but I don't know of a way to test it.
You can do this easily if you want. We just published a paper on the inspiration for astronauts, and we had them swab their skin before, during, and after the spaceflight. And it's really easy to do just with a simple skin swab. We could send you a pack, and then same thing as you've done with your stool. You can sequence the DNA and look at basically the ecosystem that's present. We also sequence the RNA of those swabs to see what's active, which bacteria are active, and
Oh, that's a great idea.
I'm pretty sure it would change a lot of your skin microbiome because we saw even just 3 days in spaceflight that the microbiome of the crew members got more similar to each other. They really started to merge into a more superorganism, at least in terms of their skin microbiome. So, and that was only 3 days in space. So I'm pretty sure if you had I don't even know what a Korean skin rub down is, but it sounds rough and
It is.
thorough.
Yeah, it's pretty
So,
intense.
if it is what it sounds like, it would almost certainly change the microbiome. But then it'd be interesting to see. It's kind of like a forest fire on your skin. So then you think about what recolonizes after this big perturbation. So it'd be interesting to study.
Oh my God. Now I want to do. All right. Well, I'm going to at least do this as a matter of personal data collection. If you're listening and you want to make this a group study, well.
Yeah, we have a protocol. We have an IRB that's active and ready. It's
Okay. So the now let's talk. I don't know, hidden. Okay, I've thrown out a bunch of to-dos on things to try. It's crazily counterintuitive that a little bit of radiation stress could actually like maybe be helpful. What especially with your study of astronauts, what's looking like it goes the other way? Like what should we be avoiding that right now we're taking for granted?
Yeah, I mean, radiation again is a balance. You don't want to have too much of the radiation, but the small burst of it doesn't appear to be, certainly it's not lethal. It doesn't seem to be a showstopper. It's a risk. It's a known risk, but we don't think it is at current radiation levels in space, a lethal risk. But the other things that go the other direction, I think one of them is actually just the isolation, like being in a small aluminum tube far away from everyone you know. That's hard enough in space, but if you think about going to Mars, there's no way to get back. There's just this conceptual, like we as a species haven't done this for hundreds of years. The closest analog is when people came to cross the ocean to look for, originally Columbus looking for India, or people, eventually settling North America, South America. It was a one-way trip. There was no way to go home. And they just had to mentally say, okay, and half of them died in some of the early years. They died on the way. The ships would crash. So the concept of that adventuresome and fearless jump into the unknown with no capacity to get back home. Some people think, oh, how could we manage that if we think about going to Mars? And I always like to say, well, we have done that before, and it's more terrifying though in space because there's no atmosphere. Like at least if you landed in North America, there was land and there was water and there was air, and there's really none of those there. There's kind of sort of land, but it's toxic. There's very little air, and there's no water basically. There's very little water, so it is a very different risk profile. But I still am confident that that isolation factor is a big one that's hard on the body. The other one is just nutrition. Like you can't grow, like when you have to take with you whatever you have. You have no ability to get a lot of diversity of foods and crops. So you can pack a lot with you, but that's another risk factor.
Yeah, it's hard to build a model of what we don't know, right? Like, if you consider the case of essential vitamins, it's not a fixed list, right? Like there have been several additions, in just this in 1 century. So who's to say that we've actually identified all the essential vitamins, right? So if you don't know what you actually need, it's really hard to like make the list of what to bring.
Yeah, and we've changed what you even need to get because, for example, folic acid is an enriched flour, but almost all flour on the world is now enriched just to avoid neural tube defects and birth defects. but it's just everywhere now. So that is a constant addition to the food supply, fluorine in the water supply, vitamin C. What's interesting about vitamin C, I talk about this in my old book called The Next 500 Years, are what would you want to engineer if you were to go into space long term? I mentioned we should be able to generate all of our own amino acids. Currently, we can't. We have essential amino acids we have to get in our diet, but the genes and the pathways that make them are known. We could reconstruct human metabolic and synthesis networks to create them. And same thing with vitamins. We have the gene that can make vitamin C inside of our bodies. It's degraded. It's old. So within mammals, most wet-nosed primates, what called strepsirrhines, like lemurs, they can make their own vitamin C. They walk around the primates. They don't need to get vitamin C from their diet. But dry-nosed primates, like us, some chimps, we cannot make our own vitamin C. But the gene is still hiding in our genome. Actually, it's still there. It's just been degraded. It's been lost over time. But so you could just modify it. Basically, you could CRISPR that, reactivate the gene. So if you're going for long-term space flight, just give yourself the vitamins that you know that you need. I mean, it's one thing to add the gene into your genome, which is a bit more dicey. But if you just kind of tweak the gene you already have and reactivate it, why not? Yeah.
Oh, I like your thinking. Wow. Thinking about the value of instrumentation, it had not occurred to me. Well, let me put it this way. We absolutely know, because of modern crises, how isolation seriously affects your physical health. So when you look at the mortality of like early European settlers in America, like surely this was a factor, but since there was no consciousness of this as a factor, like we don't have gathering of those inputs, right?
Everyone just assume, oh, that's just the cost of doing business. It's the price of the exploration. Tough luck.
And certainly it is the case. Like, it seems to me that just between the 1960s and now, we've evolved screening mechanisms in order to try to select not just like a random stochastic sample of people to send into space. So whatever you're seeing in astronauts is itself like subject to an extraordinary selection bias.
Absolutely. Yeah. And what we're doing now with like Inspiration 4, the Polaris Dawn, these all civilian missions, which are now being bankrolled by private individuals or private companies, make it so anyone can go to space. The goal is to have millions of people living and working in space within our lifetimes. And that's a fundamentally different group of people. It's going to be people that are pre-diabetic. Haley Arsenault, who just flew, she's a cancer survivor and has a prosthetic limb, and she went to space. So we'll start to see more and more, if you will, regular people that have lived and suffered by sort of ailments and injuries, and just regular people who have dad pods or regular bodies. They're going to go to space, right? So this is what's exciting, though, I think, is that we'll see a broader swath of humanity get to space, and then we'll learn about some of the health risks that we may not have known before, or maybe we'll be pleasantly surprised. Maybe they're more tolerant and adaptive than we think. So we're learning that. We're learning that right now.
Ooh, yeah. Okay. All right. So I want to ask you to speculate about something else, but before I do, I think people ask speculation questions wrong. So I'm going to share something about how we think in computer software engineering. So often when we're thinking about the design of some like extremely important thing, that maybe we want to say go fast, we characterize what bounds the performance of this program. So some programs are CPU bound, some programs are memory bound, and these are different constraints. Then once you know what the determining constraint is, then maybe you can do something about that in terms of engineering. So let's say I want to turn on my vitamin C factory, because that sounds absolutely fantastic, right? And let's just grant that there's a future where that does become a completely tractable problem. Like, oh, all right, we're going to turn this gene on. It's going to start expressing the right protein. That causes this chain of however that works. And at the other end, I get vitamin C in my body. So first of all, I'd love your speculation about like how many years out we are. But more importantly is like, when you think about trying to put a number on that, what is the constraint on progress? Does that make sense?
Yep. So good question. So I think in terms of the timeline, if we really wanted to do it, could be less than a year. Like, the reason I say that is you probably heard there's an FDA approved therapy that was approved last year for treating sickle cell disease and beta thalassemia. if you've heard about this, but basically originally published in New England Journal of Medicine about 4 years ago, the first phase trial data. What's extraordinary about it is it's a CRISPR-based therapy that treats, basically the cells in the body to reactivate a gene as a therapeutic. So the way they do it is they knock off something that suppresses fetal hemoglobin, because if there's a problem with your adult hemoglobin, you have a few options. You could do a bone marrow transplant, you could try other medicines. But what was realized is, we all, when we were fetuses, we used a different kind of hemoglobin called fetal hemoglobin. And then when you're born, though, it's turned off. And so the gene that makes fetal hemoglobin is, again, still inside your genome, still there. It's just been silenced epigenetically. It's quieted, it's turned off. But it's like a light switch. What if you just turn it back on? And that's exactly what this therapy does. It turns back on fetal hemoglobin to replace the function of the adult hemoglobin, and is a curative CRISPR therapy. And so if we wanted to just modify the glucose, the vitamin C gene, and then turn it back on, there's already a precedent that's already gone through FDA approval that we could do today. Now, the constraints are, so again, I'm saying technologically, there's different ways to do CRISPR. There's something called prime editing, there are other improved methods for this that Feng Zhang and others, David Liu and George Church, a lot of people are working on other variations of CRISPR, Jen Doudna as well. But so you want to improve the specificity, the technical part, so that can get better. But it already works today. It's already an FDA-approved therapy today. So technically, we're there already. But the constraints do you have an unmet medical need? Like right now, there are a lot of ways to get vitamin C very easily and very cheaply. And so medically, the question is there an unmet medical need? And for most phenotypes, the answer is no. Like, for example, if you wanted to temporarily boost your erythropoietin so you could climb a mountain for the weekend, like, I'm going to go to climb high elevation this weekend. I want to change my blood profile and turn on my weekend genes. It sounds cool and it would be kind of fun, but you don't really need that. You don't really, people aren't dying from it. They're just a little tired and they get, at the peak. So medically, it would probably not be approved by the FDA, but recreationally, it's something that you could technically do today. And so I think, but when you go to space, it's a forcing function. If there's that much radiation and you need to turn on your DNA repair genes or ways to prime your immune system just to survive, then that becomes a different question. So I think spaceflight is a great selection pressure of maybe we need something in addition or different than we do on Earth.
Yes, right. Then sometimes actually we see tremendous societal progress on problems of this kind just because of cost curves. For instance, when photography required very rarefied equipment, it was a very special thing. So it only happened in studios and not everyone did it. And now we have action cams that people put on their helmet, and the difference in output actually is just the cost of the inputs, right? You just drive the cost of the equipment down, and then suddenly people are like, "Well, cool, I'm going to put this on my car fender just to see how cool it is," right?
Yeah. So at some point, it's a good point. Is that the democratization of it, the access to it, really enables its broader use. It also drives down costs because it becomes a commodity. And, this hasn't happened yet with recreational CRISPRing of people doing it themselves. But there are classes you can take here in Brooklyn where you want to make a genetically modified bacteria or do CRISPR for the weekend on some trial cells. You can do it here. So it's actually where you pay $100 and you can take a class. So,
I really want to punk my children by making food glow in the dark. Can we make this happen?
Yeah, exactly. There's a company like Light Bio is one that just raised petunias that glow in the dark, for example. So
no kidding.
Yep,
I was just making that up. Oh man! Oh, that is great.
So someday soon, you could say, I want my my vegetables to glow in my fridge and to keep the lights out. Yeah.
Oh man! All right, there have been such exciting references in this interview that we're going to take some extra care to add lots of citations to the show notes here.
Oh yeah, there's a couple books, some papers I can send you. These like a lot of it'd be fun for people to see. And for further reading, see below. Yes,
yeah, exactly. Yeah, all right. But if people want to follow your research and if you feel like there's a next book in you, how do they hunt you down online? How do they follow you?
So on X, it's at Mason Lab. Mason_lab is the lab where a lot of the posts of our updates from our lab or papers we've published, work we've done is there. I'm on LinkedIn and Facebook and Insta and some of the usual social media platforms, Snapchat, and but most of it's probably through Twitter or X is where I post a lot of the most updates, and then and also the lab's in New York City, so people are welcome to stop by the lab if you're in New York. Drop me a line. You could swing by the lab and meet some of the people, see some of the modified organisms that we're making.
Cool. I'm going to take you up on that. Well, Dr. Mason, thank you. This has been an awesome talk. You blew my mind in multiple directions.
Well, my
Thanks for
pleasure.
being on the Augmented Life. Sorry.
Yeah, thank you for having me. It's a pleasure. And then I want everyone to think a little bit, to dream a bit big, because the concepts that we used to think about I think as kids seemed impossible, like going to Venus, going to Mars. We didn't have the technological capability or the launch capacity, but those things have both changed. And so I encourage the audience to think big. And the call I came from right before this one was planning out probes that we could send to Venus to look for life there. And so it's actually happening. And then other missions for Mars. So it's all happening. So dream big is my advice to any of the young ones in the audience.
Hear, hear. Thank you.